So today's class is going to be very rapid class, but very important. A lot of information will be given, and you have to be very fast to take it down. Okay, and at least 2 or 3 questions you will get from this chapter. Final. Any ionizing radiation, X-ray, right, or UV radiation. All these radiations may cause damage to the DNA. Ionizing radiation like gamma and X-rays, highly reactive oxygen particles, or ultraviolet rays. See, approximately 260 nanometer. Okay, above that it will not be harmful anymore. Right? Chemicals in the environment. Now, there are few chemicals which are naturally found in the environment which may cause DNA damage leading to mutation. Correct? If these agents can lead to initiation of cancer process, then they are called carcinogens. Otherwise, they are called only mutagens. So, the circle wise, if you see the set diagram, all carcinogens are mutagens, but all mutagens are not carcinogen. What is the test by which we determine that the carcinogen is a carcinogen? Sorry, a mutagen is only mutagen but not carcinogen. Named after Bruce Ames. It's a microbiological test. The Ames test is a test used to determine that a compound is mutagenic but not carcinogenic. Correct? Or maybe carcinogenic. So you can differentiate by this test. Fine. Now what are the other things? Aromatic hydrocarbons, including some found in the plant and microbial products. Aflatoxin, found in some kind of grass. Then chemicals used in chemotherapy. Chemotherapy is a potent carcinogen. Chemotherapy is done to kill the cancer cell. That means using poison specifically designed towards the cancer cell. Right? So they are potent carcinogens or potent mutagens. Types of DNA damage. Single base alteration. This is like a mutation, right? What is a single base? It may be transition, transversion, correct? Transition or transversion, those things, correct? Depurination, deamination, cytosine to uracil, deamination into cytosine, alkylation of the base, addition of alkyl group, methyl, ethyl group by alkylating agents, insertion and deletion of nucleotide, the whole nucleotide is deleted, that is insertion and deletion mutation. Base analog incorporation, what is that? Use of certain chemotherapeutic agents which are used as a base analog. Give one example, 5-fluorouracil, 5-fluorouracil. 2-base alteration, this is very common among us. The moment we are exposed to sunlight and the UV light in a particular wavelength, this occurs each and every day in our body, but still we do not die due to cancer. These are very common DNA damage. What happens here? Here side by side, that means 2 adjacent cytosine or thymine, they join together to form a cyclobutane ring. Cyclobutane means 4-membered ring, cyclobutane ring among the 2 adjacent cytosine or uracil to form a carcinogenic entity. Now these thymine dimer or cytosine thymine dimer cannot be recognized by DNA polymerase as a single thymine or single cytosine. They do not recognize. In their book, that template is absent. So what they do, they just simply put anything against it and continue the replication. So there is a possibility that in 25% cases it will be a mistake. Correct? Sorry, 75% cases it will be mistake, 25% it will be right out of 4 bases. Now, chain breaks when the whole chain can be broken. Strong ionizing agent. You sit inside the nuclear medicine room where we scan thyroid glands. If you are exposed to those rays for prolonged period, then the next generation will understand. Your next generation will understand. Nothing will happen to you, but it will cause damage to your gametes, which will lead to malformed next generation. Cross linkage. Sometimes 2 bases may form cross linkage, which may be read falsely by the DNA polymerase during polymerization or this thing. Now, DNA repair. From here you write down DNA repair. Always try to write down in a flowchart manner everything. Okay. So DNA repair are of 2 types: direct repair and excision of DNA repair or excision based repair. Okay. DNA excision, one is direct repair or direct reversal of damage, direct repair or direct reversal of damage, and there is excision repair. All types of this thing repair mechanism comes under these 2. Fine. What is a direct repair? So give the heading direct repair. Then write like this, flowchart. This is the only way to remember so many things in one paper. What is the major enzyme here? DNA Photolyase. So sunlight causes the damage. Sunlight itself activates DNA photolyases. Right? Now, this activation and its work may occur in dark also. That means when we are inside the classroom, we are not exposed to sunlight. When you are inside the sunlight, inside the classroom, that doesn't mean this repair work is not going on. The activation process requires sunlight. The moment it is activated, it will remain activated some time. For that period of time, it will do its work. Whether we are in the sunlight zone or we are in the shade zone, doesn't matter. Okay? So the direct repair requires sunlight doesn't mean you have to stand inside in the sunlight. Okay? You just need small amount of sunlight exposure, which will activate the DNA photolase group of enzymes. What do they do? Remove cyclobutane pyrimidine dimers. Removes cyclobutane pyrimidine dimer. Now, if this happens, what will happen? like this. Okay, in this strand, two adjacent cytosine or one cytosine and tyrosine, thymine or two thymine has formed a dimer. If the DNA polymerase reads this strand, then the daughter strand will be all right with no mistake. But when the DNA polymerase will read this strand as a template strand, then what will happen? After coming here, they will be confused which base to put. So they will put anything and go off. Most probably they will put A or G. Why? To maintain the uniform width of the DNA molecule. Understood? Uniform width of the DNA molecule. So instead of, suppose if it is T and T, then 2A should have come. But instead, there will come only 1A or 1G. So one deletion and one wrong incorporation, both the chance is there. Fine? এটা বোঝা গেল? এইটাকে ঠিক করবে কে? এটাকে ঠিক করবে ডাইরেক্ট ফটোলাইজ। কি করবে? এটা নিউক্লিওফিলিক অ্যাটাক করবে। অ্যাটাক করে এই হলো? এই ডাইমারটাকে সরিয়ে দেবে। কি আর সরাবে? এরা সরাবে না। A portion of DNA will be removed. Okay? সবচেয়ে বেশি এটাই হয় ম্যাক্সিমাম কেসেস। তো ডাইরেক্ট একটা স্ট্যান্ড রয়েছে, আরেকটা স্ট্যান্ডকে ব্যবহার করে আমরা এখানে করবো। কেন? We have this strand intact. This strand intact. Both the strands can be used as a template in replication. Fine. So it is removed and site DNA. DNA is back to normal. That's all. You should remember the name of the repair system, the major enzyme or gene responsible for repair system, and a major procedure nooks wherever it acts in special way, that special point only. That's all. The detailed process will not be asked. Even if it is asked, it will be asked as a part of the long question. So it will carry many subdivisions in the marks. So you can carry, you can score well by remembering less. Direct. Now this direct damage reversal, number 2. Direct repair by photo activation or photolysis. Number 2 is a thymine dimer is formed or an alkylated pyrimidine is formed. There are many alkylating agents like MNNG. Okay, this one you can take O6-methyl-guanine, DNA methyl transferase 1 and 2, MGMT, also called DNA alkyl transferases. Remove this. Now, if there are certain alkylated purines, then using direct dealkylating agents we can remove the alkyl group. আচ্ছা আমরা এটা আগে লিখে নাও, তারপরে ওটা এটা পরে আসছি, এটা আমি এটা পরে আছে, এপি সাইডটা পরে আছে, ওটা থাক। endogenous endogenous deorganizing এখানে ওইটার নাম দিতে পারো কোনটা MGMT তাহলে একটা অ্যালকাইলিং এজেন্টের নাম বলো MNNG একটা ডিঅ্যালকাইলিং এটা অপোজিট ডিঅ্যালকাইলিং এজেন্টের নাম বলো MGMT ট্রান্সফারেজ 1 or 2 এর পরের লাইনটা কি হবে ডিএনএ ব্যাকস টু নরমাল হলো আচ্ছা আচ্ছা এক্সিশন রিপেয়ার এর মধ্যে বিইআর একটা আছে এনইআর একটা আছে মিসম্যাচ রিপেয়ার একটা আছে আর স্ট্র্যান্ড ব্রেক লেখো স্ট্র্যান্ড ব্রেক লিখে রাখো পরে বলছি এটা SOS রেসপন্স একটা আছে আর রিকম্বিনেশনাল রিপেয়ার একটা আছে পাঁচটা মেইন ঠিক আছে আচ্ছা বেস এক্সিশন রিপেয়ার এক্সিশন মানে কি কাটা টু কাট এক্সিশন মিন্স কাট ওকে আগে এটির পার্থক্য বোঝো বেস এক্সিশন তার পরেরটা হচ্ছে নিউক্লিওটাইড এক্সিশন দুটো আলাদা কি ডিফারেন্স কি আছে প্রথমটাতে আমরা ইন ফার্স্ট ওয়ান উই রিমুভ অনলি দ্য বেসেস রাইবোজ টাইবোজকে সরাই না খালি বেসটা সরাই তাই না ইন সেকেন্ড ওয়ান উই রিমুভ দ্য হোল নিউক্লিওটাইড কারেক্ট প্রথমে আমরা এটা পড়ি প্রথমে আমরা এটা দেখি fine so there is a mistake or damage in the DNA strand correct is first recognized by certain enzymes. We are talking about nucleotide excision repair. Now, due to formation of this thymine dimer, the DNA strand will be like this. It will show a bulging, correct? So it can locate this distorted region of the DNA and find out some problem is there. So what it will do, it will remove 1, 2, 3, 4. The fourth phosphodiester linkage on 3' side and fourth on 3' side, seventh on 5' side. They will cleave. That means they will produce standard NIC. Now count how many nucleotide sequence has been separated in that NIC. Correct? Actually, how many? 10. Those two are counted as 1. So actually 10 nucleotide. So this 10 nucleotide region right, removed. Repair. See, if you are not reading the topics along with the class, problem for you, I tell you. DNA polymerase is used for repair mechanism. DNA ligase is used for joining the ends of the polynucleotide chains. So, after the DNA polymerase 1 and ligase employ, forms this. Fine. DNA backs to 3. কি মনে রাখতে হবে? পর লাইনটা তাই না? তাহলে NER is more complicated than BER. দাঁড়া, এনজাইমের নামটা হ্যাঁ, এই যে কি ডিফারেন্স? তুমি मजा आया? কি খাচ্ছ? মেলোডি? মেলোডি খাচ্ছ? বলো, what is the difference between these two? Small uvrA, capital uvrA. Next, partner in crime. Now enjoy the punishment also together. বলো, বলো না। তুই বল, লাস্ট বেঞ্চ মানে কি এটা? কে বলতে পারবে? অন করিস না, অন করিস না। খুবই লজ্জার বিষয়। I am worried about your batch. Very much. I am very much worried about your batch. ঠিক আছে, এবার রেকর্ডিং চালু কর। So, uvrABC genes are activated. They produce uvrABC product. There are 3 genes, but the product is 1. Right? The genes, 3 genes are different, but the product, ultimate product is uvrABC endonuclease. Any question? No? Fine. So uvrABC endonuclease is activated, which cleaves the disrupted area in this manner. The 4th one in the 3' end and 4th phosphodiester bond towards the 3' end and towards the, remember, this is the thing you have to remember in this question, right? Rest of the things is okay, fine. Now, base, what is added? This one, in between these 2 steps, there is one extra step. What is that? In case of base excision repair, they point out the faulty base. The base is removed by a separate enzyme called the bond between sugar and the base. What do you call that bond? Glycosidic bond, right? So the enzyme which cleaves the glycosylated bond is called glycosylases. So DNA glycosylase enzymes, they cleave the faulty bases. It can be methylated base, it can be demethylated base, it can be alkylated, improperly alkylated base, it can be damaged, broken base, it can be mismatched base like in this case. So all the repair mechanism are running on simultaneously because we don't know in which region of DNA which method will be required. Correct? So when you go out for a long trip, you take 1 rupee, 2 rupee coins, 5 rupee, 10 rupee coins, 10 rupee note, 20 rupees note, 50 rupees note, 100 rupees note and 500 rupees also. Why? Because you don't know when you are going to need 1 rupee or 2 rupee. Where will go to ask for? Right? Similarly, the cell has devised several types of repair mechanism, but it will run simultaneously. It will on its own, it will decide which to be applied when. It depends on the stage of the cell cycle where it is actually working. Here DNA glycosylases, they remove now this particular reaction, the DNA glycosylases, produce a site called AP site. AP site. AP means a purinic or a pyrimidinic site. So the endonuclease which cleaves this region are called AP endonuclease. Correct? The rest of the part, the same. Only thing is what? This counting is not correct. 4th and 7th is not maintained. 4th and 7th position, which is strictly maintained for NER, is not maintained for mismatch. Now, to understand mismatch repair, you must understand that when does it take place. These two occurs when replication is going on, rather transcription is going on. Then only they find out some problem is there and they do it. So, replication and transcription, then they are going on, ongoing process, then they point out something is the thymidine is there, AP site is there, so remove it, before rectify it before going for transcription, correct, or further replication. But what happens when replication is over? Replication is over. Now the cell is unable to understand which one is correct and which one is incorrect strand. There is no other linkage for DNA strand. So how the cell will know that which DNA strand was previously a parent DNA and which was newly formed? So which one is supposed to have mistakes? The parent DNA or the daughter DNA? Daughter DNA, because it has been synthesized later. So most probably that is synthesized with mistake. How do you know which strand between these two strands, which one is parent and daughter? Correct? It is determined by the methylation pattern. The particular pattern of methylation makes us understand or helps us understand that which strand is actually the older strand, which one is newer strand. The newer strand will have less number of methylation because less amount of time has spent. So suppose the actual DNA must have 100 methylation sites. So one strand is methylated at 100 sites. Another strand is methylated at 80 sites. That means which one is a daughter strand? The strand which lower number of methylation. So these are recognized by certain compound. Now for mismatch repair, now where do you find this particular methylation? The scientists have found after scanning the whole genome that a particular sequence called GATC they looked for how many GATC sites are present in a given DNA strand. Usually, the A site, correct? Suppose they find that this strand happen because ends are not always equal, right? But here out of 100 GATC, 80% are methylated. That means which one is newly synthesized DNA strand? The one with the lower number of methylated GATC, right? So this is how they scan the enzyme, and then take a photograph of this for flowchart. So see, they look for GATC sequence. Wherever they find GATC sequence less methylated, those strands are arbitrarily, when 80% of GATC is methylated, that means 80% is unmethylated. Among these 80% GATC, maybe between 2 GATC, one mistake is there. Rest 78 GATC are fine, it's without mistake. It is not methylated, but it is without mistake. Here, that cannot be assured. They will find any 2 non-methylated GATC sequence. Cleave it, remove it. They will not see for mismatch. They will see the mismatch is there, that will be told by some other enzyme. The GATC with non-methylated regions will be recognized by some other enzyme, and it will be cut by the enzyme which is adjacent to the second enzyme. Means the one who recognizes GATC site. So the enzyme which recognizes GATC site and cuts are not closely related to the enzyme which scan the DNA for GATC sequence without methylation group. So they may not work with a synchronized manner. In a synchronized manner, there might be some mistake, there might be some overlapping regions. So some GATC sequence between two GATC sequence, it will remove the portion of DNA, and that portion will be rebuilt, resynthesized and joined. Right? This one. Now, here, write these 3 enzymes. In this region, here, this region, defect removed by exonuclease, this region, you write these 3 enzymes, MUT-L, MUT-H, MUT-S, M-U-T, MUT. These are 3 enzymes. Again, what is the gene? Small M-U-T. What is the product? Capital M-U-T. Right? So, MUT-L, MUT-H, MUT-S. These 3. What are 3 enzyme lage? Huh? Fine? তাহলে এইখানে জাস্ট ওই এনজাইমটা লিখে দাও, এইখানটায় লিখে দিয়ে বাকিটা সেম থাকবে। ডিএনএ পলিমারেজ 1 এন্ড লাইগেজ লাস্ট স্টেপটাতে একই থাকবে সবার। লেখা আছে the underlying concept of SOS response is when DNA is being replicated, DNA polymerase 3 is going on at a very high speed, replication is taking place, high positivity. Then there is no time for DNA to wait. Why? Because the cell might die anytime. It is its earnest duty to produce copies of DNA before it dies. While doing at such a high speed replication, the DNA polymerase 3 stumbles over, হোঁচট খেয়ে দাঁড়ায় একটা জায়গায়, কোথায়? Where he finds something is wrong. What is that? Maybe some thymine dimer, some AP site. Now, it is in problem how to identify which nucleotide was or base was present there. No idea it has. So, it has possibility of choosing 1 out of 4 nucleotides, either A, T, G or C. So, it will put any one of these randomly and continue the replication. Why? Because it is better to survive with some loss than getting extinct. আমি তোমার পিছনে একটা তরোয়াল নিয়ে তাড়া করলাম, with a sword. Suppose I am chasing you. Now you are caught in my hand. Now I say you have to give some part of your body to me. You decide, will you donate your throat or your finger? What you will do? Why? Bolo? This line is important. it is better to, it is বল SOS রেসপন্সের লাইনটাই এইটা, it is, এটা আমার তৈরি করা নয়, সায়েন্টিস্ট বলে গেছে, it is better to survive with some loss than getting extinct. যদি ওটা রেপ্লিকেট না করে তাহলে সেলটাই মানে ওই ব্যাকটেরিয়াটা এক্সটিংক্ট হয়ে যাবে। তাহলে একটু একটু এনজাইম ফেনজাইম থাকবে না, একটু চুল কম থাকবে, একটা চোখ থাকবে না, একটা হাত থাকবে না, বেঁচে থাকবে, তাই না? বোঝা গেল? বাংলা একটা কথা আছে, কি আছে? নাই মামার চেয়ে কানা মামা যদি ভালো হয়, এটা কেন ভালো হবে না? কিন্তু SOS response at the same time, it is also put under the asterisk that it is an error prone repair. 75% chance যে এটা ভুল হবে। Out of 4 base, only 1 base is correct, rest 3 are wrong. So 75% chance that it will be a mistake. So it is called error prone repair. There are few enzymes are required. The error prone repair mechanism involves DNA polymerase 3, correct? কি বলেছিলাম? DNA polymerase 3 যখন রেপ্লিকেশন চলছে, ongoing process, DNA polymerase 3 and 2 other gene products encoded by UMUC and UMUD. এই দুটো জিনের প্রোডাক্ট UMUC and UMU, correct? এই দুটো প্রোডাক্ট কি করে? They on emergency basis they instruct DNA Pol 3 to insert any bases opposite to thymine dimer as DNA replication must go on with the some sort of mistake. How is the SOS system activated? Not required. দরকার নেই, চিন্তা পাওয়ার দরকার নেই, ভয় পাওয়ার দরকার নেই। অনগোয়িং এ বললাম, কি বললাম? লাস্টটা কি বললাম? ও সরি, এটা এখানে সরি মিসম্যাচ আর এটা পড়াইনি, রিকম্বিনেশনাল রিপেয়ার। লাস্টেরটা বেশ কঠিন, এটার জন্য কি লাগে? এটার জন্য কি লাগে? মাট এল, মাট এইচ, মাট এস, আর এটার জন্য রেক এ। এটার জন্য এবিসিডিও আছে, দরকার নেই, সি আর ডিটা মেন কাজে লাগে। এগুলির জন্য হলো,